I'm Jim DiCarlo. I'm a professor at MIT and part of CBMM. And what I try to prepare for you guys today is a talk that's a little bit, give some overview philosophy a bit, which I'll interleave throughout, but then also some specifics that Gabriel did a nice job of setting up around visual object recognition in the first 200 milliseconds and how we think about that from how the brain does it and how current machines do it and where the opportunities lie there at the moment. Again, the problems are not completely solved. We'll try to give you a sense of how we've proceeded so far and where we're going. But this talk is really, it's a course, so this is for you guys, so I would encourage interruption, and I'll try to encourage that along the way. I like to, again, back to a little bit of the philosophy here. So many of you here, I mean, we're talking about vision here today, but I know many of you are interested in intelligence broadly speaking. And I think a goal that many of us in this room share is, and that's why many of you are at this course, is to try to find silicon-based, what we call loosely computational intelligence that meets or exceeds biological, by which we often mean human intelligence. And so then they kind of sort of step back and say, okay, how do we actually proceed on that goal? And here's some strategies as I like to list them. You could say, forget the brain. Let's just build some stuff, and it'll eventually be intelligent by whatever operational definition we want. There's been a lot of work in that direction. I think that most of you wouldn't probably be at this course if you thought that was the completely right way to go, but it's not an unreasonable strategy. You could ignore the brain in your actual work in what you build and test, but you could talk about your systems like they're brain-inspired or brain-like. You know, companies do this for, you know, drugs and so forth, or based on science. This is really good for advertising and PR, but I actually think this is worse than just doing number one and admitting what you're doing. The third is you could use human performance as a benchmark to report your engineering progress. This is actually, some people say, well, that isn't brain-inspired because you're not measuring neurons. I disagree with that because, of course, the behavior we think rests on the neurons. This is just an indirect measurement. And so it's a little bit better because you're actually using constraints from the measurement space of the brain-performing systems of interest, in this case humans. And it's also still good for advertising and PR. But that's starting to actually be a little more of an interesting interaction between the science, the measurements of the humans in this case, and the building of your engineering. Here's another approach, and I think this is the approach that's sort of implicit from kind of classic science, and it's still, I think, debates in our field are still revolve around this item here and the one I'm going to give you in a minute. But you could just study simple reduced systems, like, okay, why a human? Why not a monkey? Well, that's not that simple. Why not a rodent or a bird or a worm? Or you could keep going down the chain and say, we're going to reduce it in the hope that principles, which whatever that means, that's usually said without definition, are discovered that are scalable. And I think the intuition of this usually comes from something like DNA. You know, our understanding, we didn't study human genome as the primary goal. You start with things like phages, and then you make progress in molecular biology, and that turns into other things. Or laws of like Newtonian mechanics, planetary motion started some of that, but it wasn't the kind of, we weren't trying to figure out all of the Newton's laws from the beginning. It was the idea that a simple system reveals something that's more general. And I think that idea is deeply imbued in how people think in science and what science is supposed to do. I'm a little skeptical about how far that will go in complex systems like the brain, but I'm just putting it out there for discussion. So the safe approach to me is essentially this one, which is you build models, what I'll call forward engineering models, within wisely chosen constraints, and that wisely chosen becomes a key part. That is wisely construing measurements, whether they're behavior, neurons, or others, and we'll talk about that in a minute. And then you care about those measurements, so you test any deviations from those models against those measurements as you go. And then if you find that there's growing deviations or that something doesn't quite match, then you adjust the engineering to respect the measurements. And that's basically an approach that I call reverse engineering. When I say reverse engineering, that's essentially what I mean. You forward engineer in the context of measurements from the system of interest. And the bonus of this approach, if it works, is that it enables not only, because you're going to converge on a kind of silicon copy, if you will, or a sort of silicon simulation of the system, that it enables advances in human health, education, brain machines, all the things that you actually want to do with this knowledge beyond just having a working intelligent agent, which again you could proceed with without the details of how it actually gets implemented in the hardware. But these things require, especially health and brain-machine interface, require knowledge of that hardware or models that are in the language of that hardware. And as a small bonus, of course, this is the greatest open problem in the history of our species. Sorry, I can barely read behind my slides here. Okay, so I'm going to come back to that slide at the end, but I'm just giving you that for the way I think about what our lab is trying to do. So when I say reverse engineering, again, another way to put this is account for each ability of the mind, which we can broadly mean intelligent behavior, using things that you can actually measure, like components of the brain. And Gabriel alluded to this too. There should be measurable, testable things. For us, this is connected neurons. In the language of engineering, and Gabriel also alluded to that when he said image computable models, so in the case of vision, but broadly predictive models. So they're falsifiable. That's one of the strengths of engineering and math, is they move it out of someone's head and onto something that can be transmitted between other humans. This is usually the domain of science, mind and brain, measurements of mind and brain, and by that I mean behavior and neurons mostly for this talk. And this is usually the language of engineering, software, hardware, robotics. And what's exciting right now is that many of this field, not all of it, but a lot of it has always been kind of interested in these neural network models. This field has suddenly gotten a lot more interested in neural network models, and that's an opportunity because these are hypothesis generators for this, which is hypothesis testers, and this interaction is becoming very, very fruitful, and I think that's partly why you're at this course. So, and it has payoffs in both directions, and I'm going to try to sort of describe for you how that's sort of worked so far in vision, what those payoffs have been so far, and for both fields, and then maybe kind of where we think about next to kind of keep this loop running. Okay, notice that we're kind of agreeing on a hypothesis space, which is essentially deep recurrent artificial neural network models. The central dogma of neuroscience is essentially that the brain, this is from Ramon Cajal, is basically a big set of connected neurons in a recurrent framework. So it's almost like, unless we're going to show that that idea is completely wrong, all of neuroscience essentially believes there's some deep recurrent neural network running in each of our heads. The question is just which one is running. This is how I think of the problem. That's what success is going to look like, is figuring out which network is running. Not, of course, there's a deep recurrent neural network, unless we throw out the entire field of neuroscience, which we could at some point, but at this point in human history, that's how it looks. Okay, so my talk today, one foundational component of visual intelligence, which we call core visual recognition. This is probably the most important slide because I'm going to show work from a lot of these folks in the lab, and I'll try to acknowledge them somewhat along the way. And again, but these are the folks that do all the work. I just get the benefit. I'm allowed to just kind of talk about how great it is, but I don't actually get to do any of the cool work. They're the ones who get should deserve all the credit. So again, my lab's reverse engineering recipe, I know I keep harping on this, but I think this is an important kind of meta message for all of you, is that first you have to specify what it is that you want to study. And I'm going to try to do that in a minute. And you want to maybe, this is where you guys can start to bring in engineering and say, you want to ask questions and say the human machine or the biological machine is in some way better than current engineered systems. This could be better in the performance sense, which is the usual way we talk about it, but it could also be power and size and other ways that you think it's better. Then you want to go and get measurements. There's measurements that exist, but you could go and get some more measurements. And here again, you have to choose wisely what to measure, and that's where some art comes in. And of course, the brain is a very complicated place. Sorry, looking for a drink here. The brain is a complicated place. And there's many things you could measure and hear about, and so it's quite confusing. And if I could tell you exactly what the right measurements were, we would only do those. I'll show you the measurements we've done and why we choose them. But there's a lot of things you could choose from. And then again, forward engineer under the constraints of those measurements. And now we say, well, okay, the models that you build should not just kind of capture the measurements, but should predict held out measurements, measurements that you haven't yet made, future measurements, or held out measurements. And then we could talk about how far away they should be held out. And that's, of course, an ongoing debate of how far some models should generalize. For most of systems neuroscience, this means that you're building artificial neural networks because systems neuroscientists tend to measure neural responses. So they want to have neurons in the models, and they measure stuff from neurons. So they want the models to predict stuff that neurons do, like respond. And that's what we do, and that sort of leads you to artificial neural networks again. And often this means because you want to predict, the models have to be built at scale. So if you're going to predict responses to new images, the models better be able to accept new images as an input. Otherwise, they're not going to make any predictions. Okay, that means image computable again in vision. Okay, and then you don't just do that and then end and say you're done. Of course, the models then sort of say, well, here's some other interesting measurements that you might want to get, or you have a family of models of which you're trying to separate and they all seem equally good or equally bad, depending how you want to look at it. Then you want to go do more experiments, and Gabriel showed this loop too, and you kind of want to go back and forth as fast as you can. And I would argue that the faster you make this go, this leads to a model which encapsulates all the measurements that you're able to get and predicts all the held out measurements until you get tired of doing that. And this constitutes, excuse me, I would argue, an understanding over the domain that you specified at the beginning. Now remember, this is maybe a narrow domain. It's not all of intelligence, but this would constitute an understanding over domain 1. Surprisingly, even this point, that this constitutes an understanding of the domain, is debated in our field. And I'll come back to that. Okay, but I'm giving you my perspective that that is what understanding is. And notice this is science. This is engineering mostly, AI, ML, CV for vision. And all I'm saying here is bring these things tightly together, make this loop run, and that's how you build understanding over this domain. Notice that these models, because the way you set them up, they have immediate or near-term application value because, again, they're predictive and they're an engineering language. They're not just textbook, quote, textbook knowledge, but they're actually useful knowledge. This is, I think, the spirit of CBMM, at least to me, around the intersection of science and engineering. And again, today I'm going to tell you about core visual object perception. Okay, so now that's kind of big introduction. Let's zoom in on the problem of visual object recognition broadly. And when I was starting up my lab in 2002, of course, you'd like to, excuse me, I'm just going to take a drink here. Oh, sorry. You'd like to really understand all of how you do scene understanding. And Gabrielle showed the Obama slide. This is this is a street scenes database slide from Tommy Poggio. We just said, let's start. You could say, well, what's in this scene? Where are the cars? Where are the people? This is my version of Gabrielle's slide. What will happen next? Where is it safe to walk? There's lots of questions you want to answer. And of course, we don't yet have systems that can do all this. But we started with just the stuff on the left here, like what's out there, and thought, can we just understand how the brain at least does that? And to start with, you start taking biological constraints. The brain, you may not know this, but your visual system has high acuity at the center of gaze. So if you look right there, you're seeing high acuity here, and it falls off rapidly as you go away from that. So that's sort of what I've tried to illustrate here. The central 10 degrees, which is what I'm going to focus on today, is about 2 hands at arm's length. Just for you to know, that's kind of the part of your visual field that I'm talking about today. That's the part of the ventral visual stream that I'll show you in a minute. Really focuses its neural resources are on that part. And Gabriel mentioned this at the end of his talk, that you don't just, the way you digest a scene is you don't just stare at one place, but you make informed judgments of the next place to look. And you make rapid eye movements called saccades. You dwell for another couple hundred milliseconds, saccade, dwell. And it's these dwell points they bring to these high acuity snapshots of that duration of approximately a couple hundred milliseconds to your ventral stream, or really your brain, to continue processing on them while you're moving on to planning the next movement in parallel. And so all that setup, that biological setup of how primates digest visual scenes like that, leads to this thing we call core recognition, which is essentially 10 degrees snapshots without pre-cueing attention and being able to judge sort of what's out there. So let me show that again. So let's watch this again. I hope that you notice that each of those are the snapshots that came out of that image that I just showed you. I hope you can notice you can recognize one or more objects in each and every one of those images. You probably could answer a question if I just showed you one of them, and I'll show you in a minute. Is a car there? Is a person there? Is a sign or tree, car or sign? And we actually test in these kind of binary modes. If you had to make a choice, what did you see? And even in those kind of durations. So this is what we call core recognition. Again, it's not all of visual intelligence. We view it as a foundational component. And here's how you test it operationally in the lab. And you might say, fixate right there. I'm going to show you an image. Here you go. OK, now you have choices. Notice I didn't pre-cue you. I didn't say what you were going to see or what you might see. I didn't say anything. And notice it was a kind of a natural-looking images. Does anybody want to make a guess? What would you say now? That was the task. Car, right? OK, here, let's try again. Okay. Oh, I wanted to remind you, why is recognition hard? I stuck this slide in. Why is this a hard problem? It's because you don't always see the car from one view. You see it, you have to distinguish cars from other things. That's usually called selectivity, in the way that tolerates or is, quote, invariant to all these other nuisance parameters in the latent generation of the image, like position, size, illumination, and so forth. This is what makes the problem hard. I think most of you know that, but I'm just reminding you of that. So we test under these kind of high variation conditions, and we can do that by either synthesizing objects where we place them in random conditions within the central 10 degrees, like here, or you take photographs, which may have some of that built in, but is less guaranteed and less controlled, but typically is, quote, natural, but depends on the data set what you use. But we tend to test both, but we've especially focused on these synthetic high variation conditions. So here we go. Here's some of these. You can try them again. OK, that's pretty easy. OK, I'm showing you kind of easy examples. OK, there you go. Bird. OK, here we go. Natural image. OK, suddenly you still know it's a bird. OK, so you can do this all day long, and you're not perfect, and we can measure that. It's actually, those were easy examples. Here's us humans. We measure us. And we knew way back then when that humans are very good at this, even at high variation conditions. That's what's shown here. This is the amount of variation in the synthetic generator. At high variation conditions, humans were swamping machines. At low variation conditions, machines were really good. That's sort of straight up pattern recognition. And you can see they're even better than humans. But this is the domain where humans were exceeding the machines in 2009. And that's where we essentially focused our efforts in these high variation conditions. OK, those are humans, but I said, well, OK, we're going to do reverse engineering. We can measure behavior. As Gabrielle mentioned, it's very hard to measure internally within the human. So we measure this species here because we can get a lot more invasive measurements. But now I need to kind of show you that species does this task also as well. There's that species going. That's the Rhesus monkey in the lab. He's sitting in his home cage. He has a little juice fluid reward tube. He's clicking a button to trigger an image, which you see in the middle test image. And then he's choosing among 2 targets just like you were. Black means he got that one wrong. Green means you can't quite see the objects that well, but I think you can see he's usually getting green squares and a little reward, which is correct. And they'll do thousands of trials like this. Again, dozens of objects that they know. It takes them about a day to learn each new object. Even though these are objects they don't actually use, like cars and so forth, they do it quite well. There was like a guitar. And here's if you measured their performance. These are the monkeys. These are monkey A and monkey B, if you like, in my language. And these ones, look, this is the sort of shows you D prime units, the absolute performance on pairwise discrimination. So these are all the different pairs of 24 different objects. There's hundreds of images in each of these squares. These are averaged over the image reviewing conditions. I don't want to dwell on that right now. I'll come back to it if we have time. But I want you to see kind of how similar these are, both in the magnitude of the D primes. You see these are some hard discriminations here, like, I don't know, knife from hanger. And some of these blue ones are easier. I don't remember. This is watch from camel or something like that. And you can see that in the D prime scale. But I want you to see how similar these patterns of confusion are. And this even holds up true at the image grain. And so the upshot of this earlier work was that this species is basically like this species and doing the task I just showed you, which is really just a statement of vision, evolution. The human visual system didn't come out of nowhere. These are a close evolutionary relative. That's why we study it. But even quantitatively, it holds up. So again, we're not next need to look for principles here. We have access to a system which has the behavior directly of interest. It doesn't need to be scaled to have the behavior. The behavior is right there in the system. Everybody with me so far? I don't need to scale up from a worm. This monkey does what a human does in this domain. Okay, everybody okay? Now you're not going to say, well, monkeys don't do everything humans. Of course they do, but in these kind of vision tasks, they're basically as good as you and I. Okay? Oh, I'm sorry. These are pairwise discrimination. So I just didn't show the other. This should say camel, 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 camel. So that's why the diagonal is blanked out. So this is how well like a camel is discriminated from a dog. So that would be camel up to dog. And that would actually be, that one looks pretty hard for it to do, which kind of makes intuitive sense. So this is just the pairwise difficulty. So there's the patterns across the pairs, and then there's just the level of the difficulty. This is sort of the performance level. Blue means really good performance. These are D prime units. A D prime unit of, oh gosh, 3 is like 90-some percent, 95% correct, or maybe 98. These are kind of psychophysics units. D prime goes up to infinity. Everybody, does that answer your question? Good? OK. Is everybody with me so far? OK, I've just basically set up another species. I'm going to tell you this equals that. And then we can come back to, well, it doesn't always, but where it does for this test of core visual recognition. OK, and that was way better than 2010 systems. OK, cool setup. Now we go in and measure this thing and see what's going on in here that isn't going on in here yet and see if we can add that in here. OK, that's sort of the big picture. And as Gabrielle mentioned, a lot of work, decades of work in neuroscience had already told us what parts of the brain were involved. It told us a lot about the architecture. We knew a lot about this monkey system, much less about this system here. So a lot of visual neurophysiology and neuroanatomy had been done that had kind of constrained things a bit. And so I'm going to tell you a bit about that. There's a set of areas called the ventral visual stream. Cortical areas V1, V2, V4, IT. Remember, V1 is fed from the LGN, which is fed from the retina. And again, this is the non-human primate, the Rhesus monkey. IT is short for infratemporal cortex. It was known that lesions in IT produce deficits in tasks like recognition tasks. And so this was already known to be an area to look for. And just to orient you, this isn't sort of the end of processing. These areas project to areas involved in decision action, around the bend to areas involved in the formation of long-term memory, among other areas of projection. So this is a subsystem within the brain, but it's not completely isolated. It has connections with lots of other things. This is my engineering drawing of the ventral stream. There's millions of neurons in each area that are schematically illustrated by these little dots here. I'm showing you schematically feed-forward flow, and things are largely feed-forward up to here into the LGN.
Then they start to become intracortically recurrent. I'm not showing you skip connections, which also exist. There's recurrence here, and then there's recurrence here as well. But I'm showing you, I'm going to focus first on the feed-forward flow, which gives the dominant first. When you flash an image up, you get this flow of information through here. But there's a recurrent connected deep network here called the ventral stream. When you show this image, it's captured by your nice camera on the front end of the system called the retina. The retina absorbs the image and kind of makes a pixelated, a nice pixel processed image of what's out there, which I've schematically illustrated as the activity of retinal ganglion cells in the back of the eye. Those are transmitted to activity in the LGN, new population activity patterns all along the stream. And these are not photographic copies. That would sort of be a waste of just moving a set of data around. Something interesting is happening as you're processing this up to these patterns of activity in IT cortex. It takes about 100 milliseconds to go from the input to the first response neurons in IT, just to give you time scale. Remember, the viewing duration during fixation is on the order of a couple hundred milliseconds. New image produces a new population pattern in IT. If I go back to the old image, you would return to the old population pattern on average. Okay, this is, and I'll say more about average, what I mean of that in a minute, or a little bit later. But the cool thing is you just can, this IT, the whole ventral stream can follow along easily at rates that are even faster than your saccadic rate. This is called rapid serial visual presentation. And you can produce these kind of IT population patterns, again, at a lag of about 100 milliseconds, easily at rates. This is something like 100 milliseconds per image. But you can go even faster than that, and that's been shown. So the system is a very robust image processing pipeline. And again, that's true in both a passive animal and an active animal. I know that question came up when Gabriel's talking. We can return to that. A lot of what I'm going to tell you appears to be completely true in a passive fixating animal, at least if they've got their eyes open looking at that dot. They can't help but their ventral stream engine being activated by the drive on the retina. This is, of course, the Rhesus monkey. I'm going to quickly take you through. This is what the monkey brain looks like when you take it out. Gabriel showed you, so I'm just going to go quickly. We know a lot about the anatomy from things like doing kind of neuroanatomy. Now you're sort of zooming in on a slice of V1. You start to see the layers here. These tiny little dots are neurons. You zoom in, see all the neurons. Here are dots. This is a Nissl stain. There's actually layered structure, if you don't know that, within the cortex. And the layers vary a little bit across the different parts of the brain, and that's how we assign one way we assign areas to be different. And so this is called an area, like area V1. I'm going to associate that with a layer in later models that I'll show you in a minute. These shows the cell bodies, and this is a different type of stain that shows not only some of the cell bodies, not all of them. You see there's many here, some of them, but also their kind of dendritic processes, which are quite complicated, and they have output axons that project locally or onto other areas, as I schematized for you earlier. This is just in case you haven't seen any neuroanatomy, and classically you go in and you put microelectrodes in neurophysiologically, and you get your electrode near a cell body just blindly until you hear some spikes. You record individual spikes, and you may sample neurons randomly in an area like V1. And of course, you can't sample all millions of them, but you sample some subset, usually hundreds to thousands, and then you try to use that to constrain models that might be explaining what's going on. So that's the big picture of how you do neurophysiology. I'm privileging the notion of spikes here. Remember back to when I said, what should you measure? I'm saying we're going to talk a lot about spikes coming out of neurons. Does anybody want to sort of say why it might be reasonable to privilege spikes over, let's say, I don't know, proteins inside cells? Gabriel and I talked about this at lunch. I don't know. We're studying vision. Anybody? Faster, right? I got to get this information I'm going to behave in 200 milliseconds. You guys were doing it. This is the only way we know that these neurons within the brain are able to transmit information at those kind of speeds. They have slower ways of transmitting, but to actually do online visual inference, this is the speedy way of transmitting information. So that doesn't mean we're going to model all the processes within an individual neuron, but we want to understand how spikes get converted to spikes, because that's how visual inference is running online. Notice this is a question about visual inference, not about like visual development or other things about the brain. So I'm just pointing that out for you to say this is not the only thing you want to study in brains, but if you want to study visual inference at the timescales at which it happens, that privileges spikes for this reason. So it's not just an arbitrary, we can record spikes, so let's do that. There's actually a reason that the spikes have a privilege. Okay, so, but of course we can record spikes, which is nice. This is now just to tell you, Gabriel showed you the filament and evanescent diagram. Where does that come from? Like, how do we draw those hierarchies of areas? Well, this comes from anatomy studies where you can sort of say there's an area of V1 and V2 based on histology, as I showed you, but also their input distributions. And this kind of, then you can work out that area V2 is above area V1. It's set higher in the hierarchy because of anatomical criteria. When you make injections in V1, they have projections mostly to layer 4, whereas if you make injections anterior grade tracers in V2, they have kind of so-called feedback projections, which are mostly in upper and lower layers. So this is not, you can see this is not symmetrically connected. And so that asymmetry, this is labeled feed-forward type, this is labeled feedback type, that asymmetry allows anatomists to put V2 above V1, and of course, most of the input is coming from the LGN onto V1, so that also sets the input. That's called higher, and then you can do this for all these areas, and there was a lot of work to study anatomically what's connected to what, and this is a reduced version of the filament and evanescent diagram that Gabriel showed you earlier, just so you know where that thing comes from. It's from a lot of neuroanatomy. This is a slide that I made for review a few years back, just to give you a sense of this at kind of a better scale of how many neurons are we talking about here. So here's the retina and LGN. Those are like 1 million dimensions, so 1 million neurons roughly in each area. You get up to V1, you have this huge expansion of the number of neurons. The colored areas are meant to be the fraction dedicated to the central 10 degrees of vision. Remember I said central 10 degrees. Notice how much hardware in V1 or V2 is dedicated to that, and that's essentially because it's way overrepresented on the retina. I mean, you process those peripheral regions, but you don't have nearly as many resources devoted to them, right? So, when you get up to IT, essentially it's really focused on the central 10 degrees. But I hope you can also see here, this is called the ventral stream, showed here, and you can see the numbers of neurons. I'm showing you the feed-forward path only here. There's around 10 million output neurons up here in IT, depending how you count the areas of IT, and you can see sort of hundreds of millions of neurons in V2 and V1. This, you heard about the dorsal stream. This is the dorsal stream drawn to scale. Okay, so the ventral stream is a lot of hardware up there dedicated to it, okay, just to kind of give you scales of things. So I see people taking pictures. There's a review right there if you want this slide. Okay, and I'm happy to share my slides after. So this processing edge of the ventral stream produces these kind of neural population codes in IT in a very rapid way, and these codes turn out to be very, very interesting and powerful. And the way we know that is from earlier work from many labs, including ours, but many labs who recorded from IT, where you can just show images. You can show images for 100 milliseconds. Now, this is what the data look like when you measure spikes, as I said earlier, out of IT with microelectrodes. Each row is a trial, and so each of these, so there's sort of 10 trials here, 10 presentations of this image. It's called a raster plot. Here's 4 arbitrary images. Notice this neural site in IT tends to spike a lot for this image and not so much for these. So there's some kind of at least image selectivity. Notice a latency of about 100 and maybe 130 milliseconds for this site. You kind of see this spike rate rise there. And that's kind of what I said earlier, around 100 milliseconds to IT, and there's some variability there. Here's another neural site, likes this image a little better, maybe these two, something different. Here's another neural site, likes these two for some reason. This is kind of data you get when you record in places like IT, and you just show bags of images like these, just to give you an example for what the primary data look like. Okay, I'm showing you this partly to get you to this point where I'm going to say I'm going to quantify these measurements of the neural activity. There's many things here, right? There's spike timing of individual millisecond scale spikes. I'm going to ignore that, and all we're going to do is average the spike rate over some time interval, 100 milliseconds, average over all repetitions that we collect. In this case, it was 10 repetitions. Often we do it with like 50 or more to get the average spike rate for this neural site for that image. So we're going to get one number, like here, 60 spikes per second, one number for each site for each image. This is just a starting point. You see, we're compressing the data, and we can later relax this and go to sort of time scale, time resolutions that are tighter than this right here. But just to kind of get everybody going, this is one way to summarize the neural response to this image. Everybody with me so far? I see some yawning. Maybe I got to start jumping around or something up here. Okay, you guys need some coffee or something? Okay, so. Okay, I'll move around over here and start doing something. So okay, we don't just record single electrodes, as I showed that example. We typically record with these Utah arrays. Gabriel also mentioned those. We implant them chronically. They record 300 neural sites at once. They're often multi-unit. We can isolate some single unit, and if the aficionados want to talk about that, we can come back to that. But about 300 spiking sites that we get at once. The animals, this is put in during a sterile surgery, and this is what the IT looks like when you sort of open up the dura there, place them in. There's one in there, and there's a feed wire coming out. We cover this all back up. The animal just has connectors on its head, bring the animal in the lab. Animal may do a task or fixate with the connectors on, and then we record the activity. And we can record stably often for many months from these arrays, so we can get a lot of data that way. And when you do these things, you end up with, you'll be able to sample like IT recordings. I kind of schematically showed you these IT population vectors, one per each image. This is an actual IT measured population vector. And again, the scale here is on 100 to 1,000 sites typically when we assemble across animals, those kind of methods that I just showed you. Recording a mean spike rate, as I showed you a minute ago, how I got to that mean, that one number. That's what's shown here. Green means high response, black means low response. This is to one test image. Again, hundreds or 1,000, one image. We don't just collect one image. Here's 8 images. You can see the vectors look a little bit different. And we collect actually thousands of images. So thousands of images, 100, maybe 1,000 neural sites, 50 repetitions to get high SNR, gives you these kind of data volumes that act as guidance for us as we try to build models. This is an example from IT. We also record from other areas, and with our collaborators record in earlier areas as well. But that's an example of IT neural data. One thing that you should know that I'm not going to take you through, but is a big setup for what I'm going to tell you next, is that if you just take linear decoders and apply them to IT, so you train on the order of 100 examples, like here's 100 examples of a car, or 100 examples of a dog or a zebra, or whatever these objects that you want to test on. And then you test on held out images of those things, again, under view variation. You're able to accurately predict both the level of the performance that the primate has. Remember, both primates. This is those behavioral data I showed you earlier. Both the performance level and the patterns just naturally come out of training those linear decoders. So essentially, this representation we think of as sort of one step away from producing those kind of behaviors that you see. Again, you don't need a lot of complicated computation here to go from this to this. And that's what I mean when I say these IT codes are special. If you do that on the retina and try that, you don't get anything at all like this in either performance or matching the behavioral pattern. So this may not be surprising to you because, of course, the brain is somehow driving the behavior of interest. But it just says, hey, this is an area where we're kind of one linear step away. It's actually not true in V4 either. So that's kind of why we've privileged IT for this task, because it's linearly able to explain this quite well. And we keep pushing those so-called decoder models to see what else we can, because all models, are right. All models are wrong. Some are useful. This one has been useful so far. It can also, let me back up. It can also, if you ask for other latent variables beyond identity, so I should have said that. That's sort of predicting whether it was a dog or a cat or a watch or shorts. If you want to ask about, hey, it's not just is it a plane or not, but about its position or its scale, these other kind of so-called latent variables, you measure human performance on those, which I'm not showing you. Linear regression out of IT is actually quite good at predicting this even on natural images. So that suggests, and if you're interested in that, you can look at this paper, that IT is not just coding identity, but a bunch of latent factors, at least many latent factors related to objects. Again, same data, just reanalyzed in a different way. Okay, this is all just to say IT is very powerful. And so that also is why, when I say it's powerful, if you go back to those 2009 or 2010 computer vision systems that weren't better than this, if they had had an IT code, they would have been as good as us, because that's the logic of what I just showed you. So if I could magically, I always used to kind of joke, maybe I shouldn't say this, if the monkey's ventral stream was on the front of the DARPA helicopter, they would be okay. They would have they would be able to do visual recognition in 2010. But they just didn't know how to build the hardware to go from the image to the IT code. That's what these data told us at the time, right? Yes.
So when you say the linear decoder can accurately predict that RDF matrix, did you mean the confusion matrix derived from your linear decoder is very similar as the RDF?
Yeah, so there's many possible confusion matrix. What the first study that we showed was that if you train on the order of 100 training examples of like cars versus not cars, and then you test on examples of cars, you're not going to be perfect, but if on average you're going to get a performance number, that average performance was the same as the monkey's average performance or the human's average performance. And if cars were, d prime of 3, and dogs were d prime of 2, or just, you get the idea. Dog versus cat. It depends how you can do a binary or 1 versus all. Then those patterns were preserved. So it wasn't like, oh, cars are really good in IT, but dogs are not, but humans are really good at dogs and not so good at cars. Things would go in the right pattern order in that sense. And that's kind of a version of a confusion matrix. In that case, it was averaged over images to just say cars versus dogs. In more recent work, we've been looking at image grain patterns, and those confusion matrices, we can start to detect slight differences from the straight up linear decoder. And this is work Kohita Carr, who's back there, is working on now, that's sort of telling us that decoders are not quite right, and it brings into recurrence to that. We can start to see differences in the image by image predictive power of those decoders. So I'm kind of telling you that we're kind of close on average, but not perfect at an image by image grain. But that's sort of more recent work that maybe if we have time, we can talk about. But then again, to us, that's sort of, okay, we need to build a better decoder. It's not like we think IT is not involved. It's just we were reading it probably in a very simple-minded way, but that was enough to kind of get things going. Does that make sense? Yeah. Yes?
When you talk about, you also try to decode from earlier layers, and if you plot like the decodability through layers, is it like you slowly emerging, or is it just suddenly in IT you have a definition of catching?
Yeah, performance generally increases as you go up. But interestingly, performance can be driven by a lot of factors, right? So what's most powerful for us is that the patterns that we were just talking about, they don't emerge, right? With enough training data, you can actually get performance up very high in these low levels, but it becomes unwieldy I kind of slid that in when I said a few hundred training examples. There's a kind of data for training argument there, right? How much training data do you need to actually then perform? And that's related to debates about training at the moment. But we were training with on the order of 100 examples. And yes, performance increases here, but also the pattern becomes much more matched when you go up here, which is kind of not surprising. The brains drive behavior, and it just says we're kind of where we thought we should be, which I mentioned earlier. Others had already sort of told us this is just a quantitative version of that. Yes?
So you had a passive fixation task as well as a same different task. Match to something.
It was kind of the task you saw here where, okay, so there's a number of studies I'm kind of lumping together when I'm talking now, so I'm trying to think out loud. This example, usually we do this in a two-way task like task that I showed you. So some of the human data was 8-way, but the data I showed you was essentially you get an image and then you make among 2 choices. Okay, and then I can do things like I can convert that to like how much I can discriminate, a zebra from a house. And these are all randomly interleaved. So you're not running zebra house for an hour. You're running zebra and house trials sort of interleaved, like the way I just showed you. It was like bird, but you might have noticed it was bird versus cat or whatever the 2 choices were. And that's what's sort of plotted here. So these are a bunch of binary tasks, we called them.
So my question here is that whenever the monkey is doing a task, whether the categorization performance is really improving or not.
Is the performance improving of the monkey, of the IT decodes when he's doing the task versus when he's not doing the task? So you decode IT either way, and you ask, how much does the performance improve? Cody, you want to answer?
There is a very small increment in decoding accuracy if you compare passive viewing and active viewing, maybe like 8 to 7% in accuracy. We typically find that the consistency is still pretty similar. Like accuracy is just overall performance of these 2 training decoders. The consistency would be like how much does that match up with the monkey performance or human performance. So consistency typically we find that they're very similar.
Yeah, so there's, as you said, there's some improvement on the order of 5%. But remember, you're already on very high performance things, so you should kind of, it always looks like a second order effect. The main effect is present. A lot of accuracy comes out with just a passively viewing it. That's what I meant by the ventral stream engine can't help but being driven by the images in states that are already kind of being able to tell you what's there, even in a passive case. Yes. Yeah, so no, so that's a great question. So we put the arrays in and we try to cover as much of IT as we can. There's some surgical limits in what areas we can hit with those arrays. And then we let the linear decoders take care of the rest of it. So they weight the neural sites according to what they need to perform. And so we don't have to optimize where we put those arrays to get these results. And I interpret that as if you're a downstream neuron, like, do I need to have a developmental sort of genes to sort of say, I've got to read from only this one part of IT if I want to do faces or cars. Or could I just take a bunch of wires that come to me, I do a little bit of learning, and then I can perform? And the latter seems to still be a possibility given these kind of data. So it's sort of like, again, what we do, you might say, well, that's an experimental limit. But remember, the brain has to get around that too somehow. That's how I think about that question. But it's a very good one, because it's not, I'm just telling you what the data say. It doesn't mean that, again, how we're trying to make inferences about how the brain works. I think we're open-minded. It could be that there's, the faces are wired up specially at birth and those kind of things, versus it has to be learned from a linear random set of them. And those two possibilities still to be, both are on the table in my opinion. Yes?
Just a quick question along that line. So if I design an artificial category where it's not, like, for instance, a cat, or I just create some random artificial images and you call it a category, train it on that, you still see something similar?
Yeah, that's a great question. We've talked about that. We have not done a lot of work like that. My guess would be that, if you show random white noise images and say, how good are you? You're not going to be good, IT's not going to be good. We kind of know things, we can already guess at things like that. But if you say, look, here's a Greeble, or, whatever kind of quote novel. I never like the word novel because there's, white noise, is that a novel object? No, it's not an object. But then to me, every object isn't actually novel because it consists of parts of other objects. But if you pick Greebles, then I think you would end up getting very similar to what you get. You would get the same result. But it raises these interesting questions about the training of the humans and the monkeys. I mentioned briefly the monkey needed on the order of a day, which is on the order of thousands trials. But how many training examples does one SVM trial correspond to a monkey trial? Like, we don't have a good mapping of that. We can make some ballpark guesses. But that's kind of where your question might go in an interesting way. We've not done that much work on that. But we would predict that it would work. But it's still to be done. I mean, work in the sense that they would still match. Does that make sense? Yeah. Yes.
Have you tried nonlinear decoders?
Yeah, actually, Gabriel was sort of one of the, the first study we did was recording without any monkey behavior, and Gabriel was involved in that. A science study I briefly showed up there. And Gabriel, you may remember, we tried a bunch of linear and nonlinear decoders, and you can correct me if I'm wrong, but the nonlinear decoders didn't seem to add that much performance value on these things when we did it at the time. We've not since, do you want to correct that, or did I say anything? We've not since tried to, better match behavior with nonlinear decoders, because if we're matching behavior with linear decoders, it's kind of what's the point? Is usually been, all right, why put a complicated model in? I don't have a gap in my behavioral data to explain, which is that question that came up earlier about the image-wise thing. Now we have some gaps, and that's what Co's working on now, is like, what's the change in the decoder? And there's different types of nonlinear decoders. And so now we feel like we have a data gap to be filled at the image grain that we can try to push on that. But that's why we haven't done that much work. But something, I don't know, it's one of the many papers Co's working on. Co, do you want to say more about that? Just briefly.
Yeah, I've been trying some models that integrate information over time to go to some point, or put an LSD on top of IT that is one other nonlinear decoding. And the formal one needs to be focused.
Right. And the thing to keep in mind, even the linear one is really good. It's just we can detect there's a problem, and then he adds this, and we can start to close the problem. Right? So you get a big bang out of just doing the straight up linear 100 milliseconds, but now we know that's probably not the right answer. It's just good enough. But great question. I'm going to move on. Oh, there's maybe one more quick one. Yeah. Yeah.
Does this representation give rise only to the rise of spike rate, or if there is some inhibition, will it also be included in this representation?
Yeah, the decoders, they're not biological in that they can have negative weights and positive weights. They don't respect Dale.
The linear model, I'm sure, but the way you represent the IT activity.
Oh, well, those are usually, we do some normalization, but they're just the sort of unit spike output responses over the, we might z-score them, so they go from negative something to positive something. But maybe I'm not understanding the question. The spike rates aren't negative when they come in, right? They're just spike counts.
It's different if a neuron had some firing rate and then had an inhibition, or just didn't fire.
Well, again, we would just take whatever spike count it elicited and put that number up for that image, and that's what ends up on the, and that's the direct. Then we might do some norm within Neuron. Again, that won't matter because you could rescale the decoder weight, but so maybe we can talk more offline if I'm missing some key detail there, but. Let me just move on. OK, I already said this. Oh, by the way, if you think that's a good model of a decoder, we call it a decoder going from IT to behavior, so it's neurons to behavior. If you think that's a good model, it sort of makes predictions like if I silence this neuron, then the behavior should shift in that way. If I silence this other group of neurons, or if I activate this group of neurons. So those are kind of like BMI type predictions, and we've started to test some of them to the extent we're able. And so far things are roughly consistent, but again, nothing's yet perfect there. But I'm not going to talk about that. But just to kind of orient you, those are models that go from IT to behavior. What I want to spend the most of the rest of whatever time I have left talking about is this thing that Gabriel also alluded to. This is like, what is your model? This is usually called an encoding model. Once we sort of fix IT here, how do you encode from the image all the way up to IT? How is it computed in this predictive sense of an artificial model that can predict the IT responses? And similarly, what are the intermediate codes that are computed along the way? Or in other words, that you want a model that matches all the way up the system here. And we had a bit of a breakthrough on this in 2012, and I'm going to tell you about that now. So these encoding models, so the idea is you should know in 2012, I showed you some images. The responses of IT neurons were a complete mystery. I think they're still mysterious, but they're less mysterious than they were. So what do I mean they're a complete mystery? I meant that you would take neural responses, and I could have plotted these for you, and I'm applying them for you now. This is the response of an IT neural site. This is one of many sites, remember. This is 1,600 images shown here. This is response. So this is not time. These are images generated of animals, boats, cars, chairs. You can see some of the chair example images here. Remember, these are synthetic images. And OK, so there was a whole, people would spend many papers trying to sort of say, well, what does this neuron do? Is it a chair neuron? Is it a plane neuron? Maybe it's neither. OK, maybe it likes some edges. There was a lot of sort of trying to guess at what makes the neuron go. And you can try it here. I mean, you can hear some example high responses. Maybe you would eventually guess at something. That really didn't get very far in the sense that those guesses weren't really predictive models. And you can see the neurons aren't really chair neurons, because if they were chair, this would be flat, high, down. They're not chair in a simple sense. There's something complicated going on. There's the images zoomed up just if you want to see. These are all high images for this neural site. If you know what makes this neuron go, good. I think you maybe could predict the future thing. I don't know. You can be drawn into reading tea leaves is another way to put this. This is the famous IT neuron. This is the so-called face neuron. It responds on average higher to faces, but not all faces. Here's some bad faces for some reason, and there's some good cars. I don't know. So it's not fair to call this a face neuron in my mind. It doesn't do the neuron justice to what it's doing, because if it was a face neuron, it would be flat, high, down. It may be involved in face processing, may be involved in processing round things also. This is just what it looks like, and you can kind of convert this to a number that says it likes faces more than other things. Therefore, it's a face neuron, which implies that it would respond high to all faces and low to other things. And that's just strictly not true. No neurons do that. Again, that doesn't mean they're not involved in face processing. It just means they're not simple in their pattern of responses. So here's an example of images. These are all high images. That's a low image. So in this case, it didn't actually like that round thing. So maybe that's some hypothesis in your head for these examples here. All I'm trying to say here is you couldn't look at these images and build some word model of what makes these neurons go. But OK, let's build some real models. Let's build some engineered models that would make some predictions. And we don't have to build those from scratch. There was a lot of neuroscience data, of which I mentioned a bunch of it along the way. We knew a lot about the anatomy. We knew that things are not fully connected here, that there was local processing in each area. We knew that even in V1 from Hubel and Wiesel, you got an oriented cell here. You got another similar oriented cell here. So there was a notion of whatever you applied at one part of the visual field, you'd apply at another part of the visual field, which today we usually implement in models through an operation called convolution. The brain doesn't do convolution, but it does parallel implementation of similar filter types, which can be implemented as a convolution. That was known decades ago. Threshold nonlinearity, certain types of normalization and pooling, the fact that we have a deep stack of layers, you don't go in one layer to get to IT, you have multiple layers, which I already introduced to you, that you have strong feed-forward flow, that you have population rate codes, that seem to be sufficient, and I show you some of the evidence for that. Again, that it takes about 100 milliseconds, which constrains the kind of, again, more of a feed-forward argument here. And we were not the first to be building models. Fukushima started in 1980 building models like this one. This is called the neocognitron, which was essentially trying to take a lot of these ideas and implement it and see if you can make something perform well in recognition tasks. And then Tommy Poggio, who's the CBMM leader, and we'll speak to you guys later in this course, they built a whole family of models called the HMAX family, which was sort of an advanced version of this and respected the biological data a lot more and made progress on predicting IT neurons and among other things. And that was called the HMAX family, and again, that went for a decade. Some of my students spent time exploring these feed-forward class of models. Dave Cox, who's now at IBM, and Nicholas Pinto, who was at Apple, they spent time building models, searching parameters of models that might better explain the biological data, and found some even higher performing systems within this large family of just feed-forward models. And then we had this breakthrough in 2012, and Dan Yeamans, who was a postdoc in the lab and Hong, a grad student. And I'll tell you about that now in case you don't know this story, because there's an important lesson. There's a model that came out of that called HMO that we don't really use anymore. So it isn't about the model, it was about the style of approaching the problem that we still kind of stick to a large degree. And the style goes like this. You have a hypothesis space that's roughly constrained by the neuroanatomy and some of the neurophysiology. So that's neuroscience that said you should have a deep, mostly feed-forward model. These are meant to be neurons in here where you apply similar operations across the model, across each layer, different types, like oriented filters, if you like. And those then are, again, applied across the areas, which is usually implemented by a convolution. And so this is called a deep convolutional neural network. This is sort of the style of model that was suggested by decades of data. And if you zoom in on this, you might have linear operators like this. You would have rectifying nonlinearities. And you might have normalization, and you can do normalization in different ways. And the way current deep networks do this is not exactly biological, but it was at least loosely biological as originally built. So I like to say these first deep CNN models were essentially sort of brain constrained, but not brain perfect matches. But they weren't just random collections of stuff to do this. A lot of the core ideas from physiology books and anatomy books were in there. But then, okay, we took that family of models, and we said optimize them to do the task that we kind of thought the thing was supposed to do, which is this core recognition task. At least that's what we thought. It was a big chunk of what the ventral stream was doing. So you optimize it to do recognition across changes in viewing parameters that I mentioned earlier. And that you could say is loosely driven by cognitive science. And then we used a bunch of engineering tricks to automate, to sort of basically find the parameters, the ones that neuroscientists didn't tell us, some of the hyperparameters and some of the weight parameters to actually achieve this optimization. So I want to kind of pause here to say the tricks that we use, I don't think were biological. I think of them as like trying to do evolution in a computer to just achieve this kind of solving these tasks under these kind of architectural spaces that you put in as essentially priors on how you build the model. And then you can ask, okay, what the breakthrough, let's get to the breakthrough, is that once you sort of set these these parameters, you end up with a model which makes predictions and can be compared against the brain. And it turns out that it matches quite well, and I'm going to show you that in a minute, that you just put in these three ingredients and you actually make a lot of progress in matching the internals of the brain in areas of like IT and V4 and other areas as well. And that's what I'm going to take you through next. But I want to pause here just so you guys can kind of understand the setup. Is everybody following me so far? Yes, okay. Maybe this is old news to you guys. But okay, what does it evolve to be? It evolves to be something that looks like the visual system. There's a deep, look, it's feed forward only. It's kind of roughly brain constrained. Here's the brain. Remember, I've been showing you schematically. These are meant to be actual neurons in here. We can measure behavior, which I told you earlier, with linear classifiers approximated on IT. But remember, what's cool to me is now people are building models with, quote, neurons in them. So that allows one-to-one comparison. So you can take the neurons out of the IT, quote, IT level of a model or the V4 level of a model. You can compare it with the actual neurons in IT and see how well does the thing match or how well does it not match. And the way we do this, and there's many ways to do this, Gabrielle mentioned this a bit. The simplest way to think of this intuitively is find the neuron, record a neuron here, and then search through here and find the neuron that looks most like the neuron that you've had here. And then use some of your neural response data to search, and then use the other neural data to ask how well it predicts on those held out data. That's sort of what point 2 is. There's details here on how you want to do this, and in fact, the way we typically do it is by taking this as we take a linear map from this whole feature set to each recorded neuron, And then once we build that linear so-called map, I heard someone say map in one of the questions earlier, using some of the neural data, we use other neural data, held out data, to ask how well that prediction system can then predict the responses of those IT neurons on those held out images. And we can do things like hold out whole categories or whole types of stimuli or just hold out images. And we've done various versions of that. For now, just think of it as held out images. Okay? It turns out when you do this, it works really well. And it's sort of, well is not perfect, but well is far better than any models that we had before. So again, and I'll show you that here. Here's this IT neuron that was mysterious to describe in words. Here's the prediction from the IT layer of what this IT neuron is supposed to do. Remember, these are predictions on these images, fit on other images. And I hope you can notice that this thing is following not perfectly, but quite nicely, that even the image grain. You can see, look, oh, there's some problem here. Okay, pretty good. Okay, there's a little problem there. You can see it. It's not perfect. It explains about half of the explainable variance when I define variance the way I showed you, mean rates over 100 millisecond windows. That was a huge advance over what we were able to do before. And it's not just this neuron. Here's the face neuron. And it kind of says, oh, I'm not just going to tell you it's a face neuron, which would be a flat square down, but it gives you all the details of what makes this neuron go. And whether you want to call it a face neuron or not is beside the point. It's a feature 12 of this model neuron in my language that would then sort of explain this neural responses. And I think Gabriel was making similar points in his talk. Again, quite good. And here's a quantification of that. Here were a bunch of old models and fitting IT. Here's the IT layer of the HMO model that I mentioned, explaining about half of the explainable variance. Turned out the internal levels of the HMO model were explaining most of the V4 variance, which is the internal level of this brain system as well. So this is kind of old news for us, but this was a breakthrough because his previously mysterious responses were now at least mostly explained. Remember, half of the explainable variance. So before it was like 10%, and now it's half. So it's still half of it to be explained, even by the ways we've measured it. But that was a big advance. So it's a half full, half empty kind of story. This is at the neural spiking level. People have done similar work in this style using these kind of models from fMRI, and I refer you to work of Nico Kriegers-Gorde, Aude Oliva, Jack Gallant, and Jitendra Malek, Justin Gardner, among many others now who've done that. Here is kind of a slide. So I'm kind of telling you, this is kind of a big picture kind of meta point that we think is important, not just for vision, but for other systems, at least other sensory systems, but perhaps other brain systems, is that I just want to give you a big perspective on what we think we did when we built this model. Remember, that model is just, it's not a final model. It was just an advance over what we had before. What we did was, do you look at this as accuracy on core recognition task, which was a kind of, you could call it an engineering goal or an AI goal. It was a particular chosen goal. Again, for us, that was inspired by what we thought the ventral stream was doing. This is a different goal, which is, can you predict the neural responses to arbitrary images? And so this is the ability to do that in variance explained units. Each dot here is a deep CNN model from that architectural family that I showed you. And what you should see here is there's a correlation. So models that are better at this are actually better at that. Here were some kind of those simpler models in the field at the time. There's that HMAX model I mentioned. And what we did, in my mind, is, OK, so neuroscience inspires this class of models. That's where the class kind of came from in the beginning. We optimize this way, again, using engineering tricks. And there's many ways to do optimization. And we used a particular strategy in that paper that led to a model we called HMO, and it was explaining about half of the variance. So there's a virtuous cycle here, which is that you kind of, again, you constrain the class, you use engineering, you then are performing well. That actually is explaining the internals, you hope, and to the extent it still does, you can keep running this kind of cycle. Now, you might ask, you could imagine you could change this to some other thing like audition, somatosensation, motor control, right? You could kind of do that with other neural networks and try to predict other internals of those systems, and that work is going on now. Though there's a problem with this, you can't only do this if you look at this for a while, because you can't just say, well, look, let's just keep building better computer vision models, if you will, and then we'll build better, it will turn into better models of the brain. And I don't have that slide in the deck, but if you're interested in continued optimization here, does not leading to continuous improvements in explaining the neural data within IT cortex. Yes.
Yeah, so I think, so the question I have is, right now you only compare the artificial neural activity in the last layer, just right before the outer layer.
I didn't mention, but we checked all layers. I just showed you in the HMO model that the last layer turned out to be the best.
Yeah, sure. But I guess my question is that right now we have deeper and deeper neural network.
Yes.
Do you think this relationship always holds true? Like always?
No, it does not hold true, and we already know that. So, and there's a whole interesting story there. We usually check all layers of all models, and I may, if I have time, I'll show you some of that. But it's not typically the last layer. It's usually somewhere in the kind of 3/4 of the way up. Okay.
And always like that. We just basically run everything and select the best.
Well, okay. So we usually check them. We check them one at a time, and then we just report the score across all of them. So you have to start worrying about you try enough models. Maybe eventually you're overfitting, but we just sweep them all usually, and then we just usually get a curve out of these models, and they're never perfect, but they usually have an intermediate peak point. Now, the interesting part is, and I don't have time to talk about this, is in Ko's work, and Gabriel mentioned it with the unrolling of the models. As deep neural networks have gotten deeper, we think what's going on is they're approximating things that are temporally unrolled, that are it gets into the temporal nature of IT. So parts of the deeper parts of the model fit the later parts of the IT response, and the shallower parts of the model fit the earlier parts of the response. That's sort of the latest trend, and you can read about that in Ko's paper a bit if you want. But I don't know, that's kind of a tangent to your question, but that's the basic answer. We generally check all levels of any model that we put up against using these kind of prediction metrics. I see a lot of hands. How much time do I have, Gabriel? When do I go till? Have a half hour. Okay, so why don't we pause? Let me just maybe see if I can get through this section, and then we'll answer questions on all of this, right? So I just want to say, again, I said about 50% match in IT. Turns out in V4 it's a little bit better, and it's gotten even better. And Puyas, who's here, could probably give you latest numbers on that. I quantify this as sort of on the order of 80%. Notice this is approximate. There's a paper here if you want to read. This is behavioral match, image grain behavioral match for the kind of tests we were doing. I always like to point out these numbers. They're not 0, which means great, but they're not 100%, which means they're not right. There's something wrong, and there's gains in them to be had. And Tolius and others have shown that things are good in V1. We have been trying to assemble a whole set of data from our lab and others that newer models can be tested on at a site called brainscore.org if you want to go there and see some of the answers to the questions. And I'll pull this up again if we have time. Because it's really a kind of field project a whole field project to come up with, find a model that can explain everything that's being observed. And we're only collecting some things. So we're trying to assemble larger and larger team-built sets in that BrainScore. Okay, let me take the questions here. Go ahead, yes. Okay.
And so how many times did you run this model with different initialization and different values?
Oh.
Does this hold?
Oh, yes. So you can run over different initializations and get nearly the same thing. And what I was going to tell you next is and maybe it's on this question, is maybe this is part of the answer to your question. Let me let you finish, and then I'll come back.
Yeah, because, one of the things that's been going on is that people are kind of, annoyed that when people build these structures, no one really does realization. There's not really one statistics right now in AI, right? You build a network, you train it, and you say, yeah, this network is great. But there's a non-zero probability that I could restart it again and get a really crappy network.
Oh, yeah.
How tight is this variance? Is this prior structure really that good? Or could you rerun it again with a different initialization and get
You have to get it to perform. What we would have said is as long as you get the thing to perform, then the match turns out to be pretty good. That's sort of what's on this slide. I told you about HMO and architecture, but the meta lesson was essentially high performance in that kind of broad architectural space leads to internal representations which are decent matches, not perfect matches. And I'm putting this up on this slide here now. HMO, that model, was not actually optimized by gradient descent, which is now what everybody does with deep networks. It was actually optimized by an architectural search. So it was like a hyperparameter search. The weights were all random weight initializations. And but newer models with SGD optimization, so again, another type of optimization, are higher performing. That's why computer vision likes them. And that higher performance generally was leading to better fits, even when you rebuild the models or retrain them, as long as you get high performance. So the lesson was high performance. I'm putting this slide up, though, to say many people have taken our work to say the brain is doing deep learning. And all we're saying is deep learning is one way to optimize a system that ends up with the end state looking reasonably like the internals of the brain, but that does not mean the brain is doing deep learning. I view this as sort of like a co-evolution thing. It somehow got it to a state where the end state point is the same, but that doesn't mean it had to arrive there by the same learning mechanism. So I just want to kind of plant that seed in your head. It's don't take this work and say, oh, this proves the brain doing. I'm not saying you, but I'm saying all of you. Yeah.
But I'm not worried about whether the brain is doing deep learning. I'm worried about how tight is the variance of your structure, right? Does your structure lead to good performance? If I run over multiple realizations.
Well, I don't know. So I'm not, again, I don't get to do any working where there's people in this room that can see, Puya, when you speak to like how many times you re-optimize AlexNet, you're going to get to sort of you're going to get to high performance system. Yeah, Puya, do you want to speak to that? So.
Numbers to give you, what is usually compared to how much of the variance is played? Okay, sure. If it's played for 9, 10%, the variance is usually two mathematics, different mathematics, T for training the network. It's usually around 5.
Okay.
Okay, so okay, I already said this approach that I'm trying to sort of say, and this I think is important for you for the whole group to think about. Again, even if you're not interested in vision, the idea that you're going to take some constraints here, you're going to constrain some architectural space like a neural network, maybe a recurrent ANN of some type, and you're going to optimize by some choices. And many people in this room are interested in the optimization rules and what the brain learns in. But the lesson we take from this is regardless of what way you do that, you can make progress on building models that are approximations of the internals of the system in a number of areas. And these are some studies that show that. I don't think we have the brain's optimization rules. We don't really know them yet, the larger field. But this idea of actually just building things is having payoffs in the same way that I'm telling you here for vision for these other systems. So you can think about that optimization approach in whatever system that you're interested in to build an understanding, let's say, at least a model. I use the word understanding. Build a model of what's going on in any kind of systems. And these are just a few people that I know that have been kind of adopting this and using it in different systems. Okay, big picture here. Remember, I said this 50%. That's what this is meant to be. Particular ANNs are now the leading models of the primate visual system. They predict the internals about halfway. They predict the behavior a little more than halfway. And these systems, as you know, are among the highest performing computer vision systems available today. And others are going to talk about that as well, so I won't say much about that. But I want to remind you, none of them passes all our tests. And I show you already 50% variance means it's a failure by one because it's not 100%. That's noise-adjusted variance. It should be 100%. It's not, so there's something wrong. But it's pretty good. Okay, so it's kind of a half-full, half-empty story. I'll just quickly sort of show you, because this came up earlier, these we can't distinguish. So at the grain of behavior I showed you earlier, the deep ANNs, at least some of them look like the primates. They pass that behavior. But if we zoom in at image grain, and I mentioned this is I'm not going to take you through the details of this, but you need a lot of behavioral data to measure the image-wise kind of patterns of confusion. And the human and monkeys still are very, very similar. But if you put up a deep net, you can kind of already see, even by eye, there's something not quite right here at the image grain, even though it's right when you average over images within a category, as I said a minute ago. And we quantified that in this paper here if you're interested. So there's still a gap somewhere between well, not any, but at least a lot of the state-of-the-art deep networks that we were testing, even the behavioral level. And so none of these models, none of them are doing what we're doing, the primate brain are doing, even in these restricted domains of core recognition, let alone the broader problems of visual intelligence. So there's still gaps to be filled in these models. And that's what I meant here by no deep ANN passes these tests. Okay. So summary so far, you can optimize models with an architectural family, get high performance, maybe the learning rule is not right, but the end state model is a big advance of the model system over models that we had before, but not yet perfect. Okay, that's a summary to where we are, and before I move on to the rest of the talk. Yes, and then, yeah.
You mentioned earlier you could linearly decode from the IT neural responses some spatial information like location and scale.
Yeah.
Have you been able to find that as well in these?
It comes out of the models too. And that's in that Nature Neuroscience 2016 paper. And that's actually, that was one of the main points of that paper, that even if you're just optimizing the models for categorization, a lot of these other latent variables come along for free. And we're not really the first to say that. Again, there's a lot of transfer work in those models. But that particular version is in those models. That question is a big part of what that paper is about. Yeah. And then I saw one in the back next. And that's Ha Hung and Yeamans are the first authors, Nature Neuroscience.
So what you said that we can't take for granted optimization is saying anything about how the brain is getting to this point. And I totally agree that you made a great prediction engine of how the brain responds and to some extent neural output. But what does this model really tell us about neurophysiology that we didn't know prior to this point?
Yeah, I think that's leading into the next part of the talk, so maybe we'll just wait for that, and then I'll set that up. But let's come back. If I don't answer it, let's come back to that question. Yeah, and then let's see. Yes.
I'm just curious, right? I understand that your goal is to explain everything, but
Well, not everything. I said wisely chosen, and then wisely I left to the beholder a bit. So, but ideally, I think all our goals are to explain everything. But yes, but sorry, go ahead.
so I just wondering how far your approach might be limited to just focusing on one task versus a very nice objective, because there might be just such representations might be more versatile in how they might be different screens, or there might be different covariances between tasks that are actually might be crucial for explaining everything.
Right. So, I mean, I think that's a good question. Let me sort of parse it this way. So, I've kind of presented to you two models. One we call the decoder model from IT to behavior. And then I think your question is very relevant because we have a particular set of things. Like I would say it generalizes over new pairs of objects, and that was related to the novel objects question, like how far does it generalize? You could call that a model. It's like somebody magically gives you IT codes for every image, and then we can make predictions of how well you will do on some space of visual tasks, of which we've only tested a subset. Right? So there I'm kind of more on your page. But for these models, remember what we're testing is any image. Now, have we hit it with white noise? And I'm going to show you a little bit of that later. But I wouldn't even use the word task because we're just showing images and predicting responses. So there is no task in the monkey training or human behavior sense of the word. There's just an image response, right? So you could say, well, what if I put the monkey to do something else? The brain's going to turn into, a black hole and something, and that came up in the active. And so that's an open-ended space of questions. But if we're just framing it as images, responses, the word task isn't to me even relevant. But generalization across images is an important question, and we think these tests are tests of that. We're going to push it a bit further on the next part of the talk here to see how far that goes. But I completely take your point with respect to the neurons related to the behavior. And you can kind of rephrase it as like, IT doesn't just do core recognition. Anyway, I kind of said it does these other latent variables, but maybe it does a bunch of other stuff that we haven't even bothered to test. So thank you. Yeah, two more.
The other 50% variance that's not explained. So in vision, or in mice vision, the recent paper was showed that the response is always as high dimensional as possible. They showed the 2,000 something images.
Yeah.
And we also know that in deep neural networks, it always land in some lower dimensional space so that it still have a generalization performance, even though it has it's in such an overfitting regime. Like, have you looked at any of the distribution of responses, or a more higher structure of the?
So I think you're asking interesting questions about dimensionality. It sounds like to me, I didn't quite parse everything you said, but the dimensional match between IT and the models is still a work in progress. Okay. And I would give you, this is an active area I've worked in the lab really for the last 5 years. Everybody is tired of me talking about dimensionality because the measure I showed you, which is a regression metric where you can take a model set and then regress the neurons, notice there's a way to cheat there. You can have a high dimensional model fitting a low dimensional set. So and I think some of that's actually going on. So this comes down to how you match things, right? So we're now trying to match dimensionality. The models, I think, are over-dimensional relative to the brain, which, but that's based on some data that we have. And so that's ongoing work to say, are we sure they're over-dimensional? How do we measure neurons? And many ways to measure dimensionality. There's many types of dimensionality. All I'm going to say to you is that if you want to talk about projects in that space, you reach out to Ko or Puya, and there's a lot of stuff in our lab trying to engage on that space of question. That may not have been all your question, but maybe can we leave it at that so I can get to the rest part of the talk. Okay, one more quick, and then.
I'm wondering about adversarial images.
Oh, maybe.
So one of the strongest challenges to this correspondence is the fact that the artificial models respond. You know where I'm going. Have you considered recording the neural responses to adversarial images?
Yeah. We talk about that experiment all the time. We almost don't think it's worth doing because we're pretty sure the neurons will be robust to that, but we should just do it to show, and then how much robustness. It actually came up with a grad student last week. Let's just run that experiment. But we haven't done it yet. We haven't recorded designed adversarial images. But I'll show you some stuff we have done in Ko's work that was on recurrence that was mentioned that I don't have time to talk about. We searched for images that were hard for models. And then we just took them, they could be, but they were easy for humans. And so you could call them discovered adversarial, but they weren't pixel optimized adversarial attack. They were just here's a picture of a dog that
AlexNet's getting wrong for some reason, but all humans just easily get it right. And
but wouldn't you actually need the opposite result? That if CNNs are a strong correspondence to the human brain, then wouldn't you need to show in some sense that the images that fool a model, the way that the pattern of fooling the model can be decoded in the...
Yeah, you're pushing on like how good of a model is this of the system, right? And let me go to the next part of the talk and then see, and that some of that comes up there. And again, I keep trying to say, like, I mean, the models are better than before, but not correct. So that not correct part is what all you guys should be thinking about. And maybe I can give you some inspiration or constraints, and BrainScore can give you some real constraints. But I'm not trying to say these models are done and correct, but I'm trying to give you some perspective that they're way better than whatever models, which were words in people's heads or sort of models, that any quantity, even the implemented models before, they're way better than any of the implemented models. So in that sense, they can't be ignored as kind of hypothesis starting points. But that doesn't mean they're right. They're certainly wrong, right? Is that okay? So let me sort of come back to this. Okay, I'm going to say I'm not going to tell you. Of course, I was talking about this two-way street. These models, as you know, have kind of taken over a lot of kind of engineering. 2012, there's like deep learning takes off, and of course now deep learning is well beyond vision, and it goes to all kinds of problems. And it was nice, at least they said, using artificial intelligence techniques inspired by theories about how the brain recognizes patterns, right? So there's at least a nod to neuroscience in 2012 when this stuff started to really take off, when these models started to win the ImageNet challenge. And I think we'll let others tell the rest of that story and where they stand. I want to talk about what these models are now doing in terms of informing our, quote, understanding of the brain, which relates to this question and the one in the back and several others. So again, there's stories to be told here, but I'm going to talk about this flow back. Now we have a model. What can it do to help us further our understanding of the brain? So, these are the best matching. Remember, none are perfect, but you can ask the question, are the best matching deep ANN, whether they're recurrent or not, models and understanding, quote, understanding of brain function, do these constitute an understanding? Because we usually hear they're a black box, so therefore they're not an understanding. And I always have a hard time with that statement because they're not a black box. We know every detail about it. We just have a hard time expressing it in ways that humans are usually happy with. But anyway, that's the setup, is like that doesn't count as an understanding. Remember way back to my beginning slide when I said I'm going to optimize, I'm going to get a model, and I'm going to call it an understanding. And so here we are, there's optimization going on, it's not done, but there's a model, and does it count as an understanding? Well, rather than debate this, we just sort of said, the way we are taking this on in the lab is we say, look, what do we even want understanding for? There's making academics happy and papers and Nobel Prizes or whatever, but then there's the work of the human species. And why do we even need models in the first place? What are we going to do with the understanding? Like, let's just make it a practical question. And I would argue that the reason anybody does science is control. So the reason that we build models, we want to fix the brain. That means we want to know what drug to give. We want to control it, move it to another state. We want to know where Mars is going to be so we can land a rocket on it. So that's a prediction. We want to know something about physics or DNA code so that we can edit it or make a new one, right? These are the reasons that we have models, is they enable control of the world. So now you can ground this into like, what control are we after? What do these models give? Well, these models, and so now I'm going to talk about Puya and Co. are both right there actually. We released this paper this year. This was like a best paper award last year in 2018. And we basically said, look, let's just do, these models say they're models from images to neurons, so let's use them to control the neuron. Like that was the setup. Now you'll see, this is very similar to some of the things that Gabrielle presented in the cool work with Marge on the kind of face neurons and what images you can find. But we took a different spin on it, which was essentially right from the outset, let's test the model and sort of, let's assume it's a good model of the system and ask it to control the neuron. So here's a model. This is one particular model. This happens to be AlexNet. And we map it to some neuron that we record, like I showed you. So there's some features here, linear map to a neuron that might be in V4 in this case. I'll show you in a minute. But then because it's a model and it's not a black box, we have full access to it, unlike this where we don't have full access. This is not a black box. We know all the details of this model. Because we know all the details, we can actually optimize on the image space to find pixel combinations that put the neuron or the set of neurons into whatever state I declare I want them to be in. So I'm saying my goal is to control a V4 neuron. I want to make it go up. I want to make it go down. I want to make it go left. I want to make it go right. Now there's no left or right for neurons. They just go up and down. But a set of them go, one neuron go on, others go off, right? So I'm setting up a control phrasing to say the reason you might want to model is maybe you want to control V4. Now we can start to think ahead, like, why do I want to control the brain? What areas do I want to control? But let's just take it as a goal. It's like, this is what models are supposed to do. If they're correct, they should at least enable such a thing. So this removes it from debates about simplicity or beauty, or is it a black box, and just sort of makes it a kind of practical thing of how well does it control the darn neuron. Is everybody with me so far? Okay, we can debate understanding, but at least we can sort of agree on that control is a test of understanding. Is that okay? So turns out you can actually do quite well. And I'll show you how we do that. So the goal here, and this is, again, it's Puya and Ko's work, and this, if you like to read about it, it's here. Guys, correct me if I say anything you're not happy with. A desired internal neural state of a target region of the brain is a goal. Given that humans decide they want to do that, they want to control V4 in this case, automated design of control images enabled by a model. So now you not just need a model, but you need methods to optimize the pixels to decide how you want to control the neuron. Notice that this would not be possible without the model. So the understanding here depends on the accuracy of this model, and what I'm going to call the understanding embedded inside the model that we are just extracting here, digesting, to get an image to do our bidding as a human species to control this neuron with tools that we have at our disposal called photon projectors on the eyes, i.e. show images to the animal. Right? So we have a lot of commodity hardware to do this. We can all pull out our phones and get controlled right now if we wanted to. So let's try to actually control. I'm controlling you with this image right here to some degree. So we experimentally assess the ability of non-invasively controlled neural states deep in the brain via these kind of externally luminous power on the retina. We just basically compute an image and try to turn neurons into whatever state we want. It works pretty well. Here's how it goes. You have model predicted firing rate. This is actual measured neural firing rate. This is one V4 site. These are a bunch of natural images. There's one. This is the best image out of this set of 640 or so that were tested. There's the receptive field of the neuron. Zoom in on it. Maybe it's something about this plane edge. Again, we could guess at it, but that's not really the way we would go. But notice how good these points are on line. Isn't that cool? Look, the model's predicting the measured firing rate. But that's the story I already told you, right? That's essentially this PNAS paper, what I said earlier. The models are really good. This is what good looks like, right? So you can actually control these neurons. Not control, sorry. You predict them quite well. These are predictions on held out images. And so the models are good. That's basically the story here before. Okay, but now we're kind of going to say, let's move this beyond that. Like, if we can control this for the single neuron, let's drive it up, maybe drive it down. Okay, but for now, it's like, let's just drive it up. Can we do that? And so can we move it beyond its natural range? That's the way we were thinking about this. That means find images that the model says, you go up here, and then this will be up here, right? So I'm going to tell you what to do, and I'm going to get up to there, and then do that. Okay, so this is from Puya gave me these nice videos of him optimizing for 5 different neural sites. And you start with some pixel seed point. And what's kind of cool, these are 5 different V4 sites. Notice they're kind of visually different, which is kind of cool in a way. They all look like textures in a way, and there's different constraints you can use on these optimizations, and he can tell you more about that. But here's the results that is that this particular neuron, here's 5 different initializations. Again, they're kind of perceptually similar, but you can see pixel-wise they're obviously very different images, but perceptually they're similar. And look, it's pretty good. But again, I keep telling these half-full, half-empty stories. Look, it's above where the neuron had ever been before across all the images that we had tested. But it's not on the diagonal, so there's something wrong with the model too, right? So again, half-full, half-empty enables new control, but not perfect, which is kind of consistent with the model that's only 50% correct, right? So, and we actually showed in this paper, better accurate prediction models lead to better control in this generation method, which when you think about it, isn't that surprising, but it actually is, it would suggest that if we build better models, then we get even better control. And we can now just design images that will make the neurons do what we want individually. But this is what gets me most excited, is not just individually, but think about a whole V4 population. And what if we want to turn on one V4 neuron and turn all the others off or keep them off? This is a V4 population. This is 40 recorded sites. This is a different control goal that we call set the population state. The neurons are recorded. They have highly overlapping receptive fields. So this is not a trivial task of injecting energy into one part of the visual field. That's what's shown here. If you take the best image that you searched through the natural images that best activated this target neuron but not the other neurons, this was the best you could do. It's pretty good. A lot of neurons are on, but this one's on the most, so it's not a bad choice among the set of just random tested natural images. But if you then kind of say, okay, you got all this undesired off-target activity, the model designed an image that said, this is the image you should use to achieve that goal, and here's how well it did. It's not perfect, but it's way better than this. So it comes up with this crazy high frequency thing that I don't know, maybe it looks like the edge of those chairs. I don't know exactly what it's doing, but it's found an image which is much better at the goal that we set up, which is control this whole population now. But again, not perfect, but better than we could by natural images. And we describe our statistics on how often we can do that and how well we can do that. This is actually a particularly strong, good example. But if you want to read the details, you can see them in this paper. So I start dreaming about, can we actually use these models to start to inject energy to, remember IT projects to the amygdala, right? So I start thinking about, okay, could we start to use these to custom design images to drive the brain into different states? This is what you'd use models for, at least one thing that you can use them for. Even with hardware we have available today, i.e. images that you can show that are, remember, this is a vast space of possible images. And Gabrielle sort of mentioned this when the grad students, how many grad students you'd have to record, or you phrase something like that, right? So there's a huge space of unexplored images that the models can help us navigate and maybe find us images to put the brain in states that we may want it to be in. Again, we want to think about this as a tool for good. We could also think of things that could be used for bad. Okay, so I'm going to try to sort of wrap up here. That's sort of one example of it to test of the model has some powerful understanding. Again, no model passes all our tests so far. Where are we going from here? Okay, if we build better models, we might be able to have better control. Well, so the race is on to discover even more accurate neural network models of the primate visual system. This is what I think our bread and butter job is to actually keep this loop running. And I hope many of you might be part of this broader we if you'd like to be into this space. And our strategy, and this is exactly what Gabrielle said, is you use the best models to drive experiments. The goal is for the experiments to break the models. They've already broken them to some degree. We can try to break them harder. Then you use those breaks to design better models, and then you keep iterating on this loop, and then you build better and better models that enable things like better and better control. One example, this is again Ko's work on recurrence that was mentioned a few times in Gabrielle's talk. And again, I don't have time to take you through this, but we've already found that these models, I've been focusing on the feed forward parts because that's how the models were initially. We can now see that the recurrence is causally important, or at least I can't say causal. We can say the recurrence appears to be important, and we have some evidence of causality. And so now we're building models that have recurrence in them as the next class of models, and that was mentioned as well. And if you'd like to read about some of that, you can read that in this paper. But that's, to me, just one example of like the next version of now a deep recurrent model, and that's how we're going forward here, is to sort of use the data to find new models. So let me, in the last 2 minutes I have here, give you some summary points. I have a couple minutes left to my, quarter of? Is that right? Yeah, okay. But maybe before I do the summary points, does anybody want to complain about the control stuff? Or say something, I don't know if or maybe let me say the summary points and we'll come back to, because I'll just summarize for what I tell you today, and then we can talk for as long as Gabrielle lets me stay up here. Okay, summary of what I tried to tell you guys. If you were asleep for the whole thing, here's what I tried to tell you. The ventral visual stream produces an IT neural population representation that carries linearly decodable, image generalizable solutions. That is, you can build a decoder and it generalizes to new images for all tested. Somebody asked about, did you test X, Y, or Z? And for everything we've tested so far, core recognition tests, of which we test on the orders of dozens of tasks, and I've showed you much of that. The second main point that I told you is when you optimize deep artificial neural network architectures, even feed-forward architectures, that were approximately in the architectural space of the ventral stream, for core recognition tasks, this leads to internal neural representations in those ANNs that are remarkably similar, but not perfectly similar, to the internal neural representations of the ventral stream. And that work was originally here in those publications. This result above includes face neurons. You just naturally get those as well, and I showed you that. This result above is consistent with, but does not imply that the brain learns by classic backprop or SGD. Again, I said this already. My preferred interpretation is that this is a demonstration of convergent evolution development. Okay, species A, primate vision optimized by evolution postnatal development. Species B, deep ANNs optimized by engineers. And those have converged to be similar but not identical, but it does not say that they're using the same learning rules. Okay, third message. These same optimized ANN models can be used to guide the construction of novel synthetic images to superactivate ventral stream neurons. I showed you before. Gabrielle showed you some nice work in IT. And to control subpopulations of neurons. And that was what I showed you about V4, turning one neuron on and others off. Nevertheless, these same optimized ANNs are not yet identical to the ventral stream. I've said this a few times already. Here's a couple of papers that sort of show that quantitatively. One difference is the lack of recurrent circuits, and recurrent IT neurophysiology. Recent IT physiology suggests that fast-acting, automatically evoked recurrent circuits enable the ventral stream's superior performance on many images. That's Ko's work that I didn't have time to talk about, but you guys can read about it in this paper here. We and our collaborators, and this was mentioned in Gabriel's talk, have built models that include local recurrent and skip connections. We call these core nets. This is our first attempt to build models, and we show that they're a better match to the IT data and are still high performing in the computer vision sense. So if you'd like to read about that. So the only slides I had left was kind of motivating more of a discussion before we get to the big questions of like, okay, what's missing, right? I said, let's build some new models. And again, I want you guys to be forward-looking. I kind of try to give you some perspective on where we came from, but you should all be the sort of leaders of the future to say, what do we need to put in these models that we don't have yet? We've already talked about recurrence at local short time scales, but there's larger time scales to expand to here, and I know Gabriel and many others are interested in that. I only talked about the ventral stream. It's connected to many areas that I loosely mentioned, many that I did not mention, and those might be important to actually building even better, more explanatory power. The brain as a whole is a big recurrent neural network. We talked about the dimensionality of the codes not yet being correct, so that's something we're actively working on. Spatial organization of the tissue. Many of you who know about fMRI or the things that Gabriel mentioned, face patches. Neurons are not randomly arranged, so we've been working on kind of what is that spatial structure in the ventral stream. Kind of, is it important? What is it for? We think it's having to do with wiring costs and energetics mostly, but that's an ongoing area of like improving models. These neurons are like loose approximations of real neurons. I showed you neurons with dendrites and crazy structures, but these are basically dot products with a threshold after them, and that's not how a real neuron works. Maybe we need a better approximation. Again, these are guesses. Better approximations of the ANN birth state. The initializations of these models we'd often think of as like the birth-childhood state. That's probably not correct. Maybe we need a better approximation of that. Again, these are just ideas. Better approximation of the learning rule. I mentioned this a couple times, which is roughly supposed to be analogous to postnatal development. There's no spikes in here. These are analog models. Some people think that's going to be important for computation. My bias is to think that's mostly important for energetics, but okay, that's something obviously different from the brain. No cell types in here, or just a couple if you like. Obviously, there are different cell types in the brain. There's lots of other things in the brain that aren't in here, synapses, channels, so forth. Lots of stuff we could put on. I kind of put these in the order of kind of my rough guesses of what we should be working on to improve this for the problems that I showed you, but those are just admittedly speculation at this point. The meta point for me is these are all recurrent ANN. No matter what we do here, we're going to have a recurrent ANN unless we kind of, again, change the central dogma of neuroscience. So this is really a search through recurrent ANN space and more approximate models of recurrent ANNs. And just coming back big picture, I'm talking about the first 200 milliseconds of post-saccadic processing. Gabriel mentioned, okay, we got all these saccades, all these other tasks. Somebody asked him a question over here about how does that answer the Obama question. I've not spoken to any of that at all. I'm just like, what does the brain do for those first couple hundred milliseconds? And I've told you where we are on that. There's, of course, many other bigger questions of visual intelligence that we strive for, but we're not yet answering it. I think Gabriel's closer to that than we are at the moment. Again, we're down in the hardware of the neuroscience. So I hopefully give you a sense of at least that problem and where we know about what the brain does on that. And I'll come back to, like, this was a slide I showed at the beginning. If you actually want to do this, not just as a kind of fun intro slide, I mentioned BrainScore with Martin Shrimp, a grad student runs in the lab. If you want to go check it out and get a bunch of metrics on how we're actually trying to make real on this kind of multi-constrained search space and adjusting models as you go, I would go there. So with that, I'm going to skip through that. And just, this is all BrainScore stuff if you guys want to hear more about that. And just end with that. OK, thank you.